muscle contraction is the most familiar and the best understood example of movement. In animals, for example, running, swimming, flying, they all depend upon rapid contraction of muscle fibers, and this process, as we know, consumes ATP. So let's look at this process in more detail. We have talked about it more indirectly, relatively indirectly. Now let's look at it in more detail. First of all, muscle cells are highly specialized for rapid and efficient contraction. The long, thin muscle fibers of skeletal muscle are actually huge single cells that form during development by fusion of many separate cells. These huge multinucleated cells form by the fusion of muscle cell precursor cells, which, as I mentioned, are referred to as myoblasts. In adult human, a muscle cell is typically 15 micron in diameter. So this is again a more magnified view of muscle cell. This is one muscle cell. It has many nuclei because it was formed by fusion of smaller cells called the myoblasts. And it is 15 micron in diameter, and it can be up to several centimeters long. Many nuclei of the contributing cells are retained in this large cell, which lie just beneath the plasma membrane, but the bulk of the cytoplasm inside the cell is made up of these myofibrils that we have talked about, which is also the basic contractile element of a muscle cell. A myofibril, as I've said, is cylindrical structure, 1 to 2 micron in diameter, and is often as long as the muscle cell itself. And because of this arrangement of the sarcomere, the muscle cell looks striated or with stripes. Each sarcomere is formed by precisely ordered array of parallel and partly overlapping thick and thin filaments, as we have seen in a previous diagram. The thin filaments are composed of actin and associated proteins, and they're attached at their plus ends to these Z discs. These are the Z discs. These cap these Z discs are at the end of each sarcomere. The capped minus end of filaments extend towards the middle of the sarcomere, which we have seen earlier, where they overlap the thick filaments. The bipolar assemblies, which are formed by a specific isoform of myosin, the myosin 2, which is present in the muscle. Now, let me show you a cross-section. So the cross-section will be taken here, so you can see the arrangement of these different filaments from a cross-section. Here is myosin and actin filaments in an insect muscle cell. So this is a cross-section. You can see that these are the thick filaments, the myosin, and the thin dots are the actin filaments. And they're regularly arranged in hexagonal lattice with actin filaments evenly spaced between them. Sarcomere shortening, again, is caused by myosin filaments sliding past the actin thin filaments with no change in length of either type of filament. So this is important. The filaments are not getting shorter. They're just sliding against each other. The arrangement of these filaments is slightly different in the vertebrate muscle. The bipolar thick filaments, when they walk towards the plus end, the two sets of filaments of opposing orientation driven by hundreds of independent myosin heads that are positioned to interact with each thin filament. When they interact, the heads, they result in coming together closer of these thin filaments, resulting in contraction. In course of rapid contraction, the myosin actin filaments pass one another at rates of up to 15 microns a second and enable sarcomere to shorten by 10% of its length and in less than 1/50th of a second. There are animals, for example, which I will show you in this slide. You have seen hummingbirds. These hummingbirds can flap their wings 50 times per second, which is fairly rapid. And this results in flying of this animal, makes this animal capable of flying. And this shortening allows us to move around walk, and other animals to swim.